The overall goal of this procedure is to generate tissue-engineered vessel grafts that are functional for grafting into mice. This is accomplished by first reprogramming human fibroblasts into partially induced pluripotent stem cells in vitro, which are capable of differentiation into endothelial and smooth muscle cells. An aorta is then removed from a sacrificed mouse and decellularized with sodium dodecyl sulfate or SDS solution to prepare the decellularized aorta grafts. Next, the bioreactor flow circuit is set up for dual seeding of partially induced pluripotent stem cells. The final step is grafting of the double-seeded partially induced pluripotent stem cell graft into mice. Ultimately, the analysis of mice mortality rates 3 weeks post-grafting is used to show the functionality of the tissue-engineered vessel grafts.
The implications of this technique extend towards the therapy of cardiovascular diseases because it has been shown to successfully generate functional blood vessels using PiPS cells in a short period of time.
In general, it's very important to keep the vessel graft intact and sterile during the whole procedure, especially for people new to this method.
Begin this procedure with reprogramming of human fibroblasts into partially induced pluripotent stem cells or PiPS cells by transfecting the cells with 4 micrograms of linearized OSKM plasmid using a human dermal fibroblast nuclear